In this presentation, we will review the structure of a generalized neuron, and demonstrate the events that take place when an axon generates and conducts an action potential. Review the structure of a generalized neuron. I've included the information from the Nervous System 1 lecture organization and histology pertaining to the structure of neurons below. Neurons or nerve cells are extremely structurally and functionally specialized. They function to generate and transmit messages in the form of nerve impulses or action potentials from one part of the body to another. In the picture above there is a single blue neuron and part of a purple neuron that it is making contact with. The neuron sending the message is referred to as the presynaptic cell, and the neuron or other effector is called the postsynaptic cell. Recall that there are many structural types of neurons, but all types consist of three main parts. Neurons consist of three main parts. 1. Cell body or pericryon or soma contains the nucleus and organelles. It contains most of the typical cellular organelles, which include the nucleus, cytoplasm, Golgi apparatus, mitochondria, and cytoskeletal elements. Neurofilaments and neurotubules, etc. The row of neurons is well developed because it is active in the synthesis of neurotransmitters. It is easily visible in the light microscope and is referred to as Nissl bodies or chromophilic substance. 2. Dendrites are neuronal processes that receive input. Dendrites are typically numerous and highly branched with dendritic spines where they make contact with other neurons. Dendrites can form arborizations, thereby increasing receptive area. Makes it possible for one neuron to receive and integrate information coming to it from the axons of numerous other neurons. Neurons receive information from other neurons via dendritic spines, which make up 80–90% of the neuron's surface area. 3. Axon or nerve fiber sends impulses. Neurons have a single, sometimes very long axon. The axon is capable of generating and transmitting an electrical impulse action potential to the postsynaptic cell. Because of their complex structure and specialized function of the axon cell membrane, a separate set of terminology is applied to different parts of the axon. Axoplasm, cytoplasm of an axon; axolemma, plasmalemma; axon hillock, a small raised area between the cell body and the beginning of an axon; initial segment, located just beyond the axon hillock. It is the region of the neuron that generates the action potential. Neurons form connections between their effectors called synapses. A synapse is an electrical or chemical junction or point of close contact between two neurons, or between a neuron and an effector cell. Three main types of synapses are shown above. In this lecture, we will be concerned mainly with neuron-to-neuron synapses. However, when we study muscle physiology, we will emphasize the neuromuscular junction, and neuroglandular synapses are important in endocrine physiology in the autonomic nervous system. Neurons are highly irritable, which means they are responsive to stimuli. A stimulus is a change in the environment. When a neuron is adequately stimulated, an electrical impulse is generated and conducted along the length of its axon. This response is called the action potential or nerve impulse. It is always the same, regardless of the source or type of stimulus, and it underlies virtually all functional activities of the nervous system. The goals of this lecture series are to explain how neurons become stimulated, either excited or inhibited, and how they communicate with other neurons or effectors. Before we can describe how neurons respond to stimuli by generating and propagating a nerve impulse or action potential, we first need to define some basic principles and definitions of electricity, and revisit the resting membrane potential described in the previous learning module. First, the human body is overall electrically neutral because it has the same number of positive and negative charges. However, there are areas where one type of charge predominates, making such regions positively or negatively charged. Because opposite charges attract each other, work in the form of energy must be done to keep them apart. Recall the cell membrane, where energy is expended to keep the membrane negative on the inside and positive on the outside. This charge differential represents potential energy for work. When opposite charges are allowed to come together, this liberates energy that can be used to do work. Where there are electrical charges of opposite sign that are kept separated, th. At system has potential energy. The following slides provide some definitions used when describing electricity. The first is voltage. Voltage is the measure of potential energy generated by separated charges. It is measured in either volts or millivolts. 1 millivolt equals 0.001 V. Voltage is always measured between 2 points and is called the potential difference or just potential. The greater the difference in charge between the 2 points, the higher the voltage. The flow of electrical charge from one point to another is called a current, and it can be used to do work, for example, power a flashlight or iPad. The amount of charge that moves between the 2 points depends on two factors, the voltage and the resistance. Resistance is the hindrance of charge flow caused by the substances or materials through which the charge must pass. Substances with high electrical resistance are called insulators, those with low resistance are called conductors. In the case of living cells such as neurons, the hindrance to flow of charge is the plasma membrane or axonema. Ohm's law explains the relationship between current I, voltage V and resistance R. The greater the voltage, i.e. the potential difference, the greater the current. Current is inversely related to resistance, the greater the resistance, the smaller the current. In the body, electrical currents are the flow of ions across cellular membranes. Recall that there is a slight difference in the numbers of positive and negative ions on the two sides of the plasma membrane. Thus, there is a separation of charge, so there is a potential across the membrane. The potential difference across a cell membrane is called the transmembrane potential. The transmembrane potential is the result of an unequal distribution of charge across a cell membrane in which the inside membrane is slightly negative due to a slight excess of negatively charged ions and proteins, and the outside is slightly positive due to a slight excess of positively charged ions. This is due to differences in membrane permeability to various ions and active transport, and is characteristic of all living cells. Recall that plasma membranes have a variety of membrane proteins embedded within the two lipid layers. Many of these are ion channels. Most have a region that forms a molecular gate that changes shape to open and close the channel in response to specific signals. These are called gated channels. Some channels are chemically gated. In this case, the protein channels has a specific area that acts as a receptor binding site for a specific type of ligand. When the appropriate ligand binds to the receptor on the channel protein, the shape change causes the channel to open and allow movement of the material to be transported. In example above, sodium and potassium ions are being transported simultaneously but in opposite directions. This particular chemically gated ion channel is an antiport. Other channels are voltage gated as in figure B. Voltage gated channels open and close in response to changes in the membrane potential. Mechanically gated channels open in response to physical deformation of the receptor. Sensory receptors for touch and pressure such as Meissner's and Pacinian corpuscles found in the integument, are examples of mechanically gated channels. Leakage or non-gated channels do not respond to signals and are always open, however they are selective as to the type of ion is allowed to pass through. Examples include potassium and sodium ion leakage channels. When gated ion channels are open, ions diffuse quickly across the membrane following their electrochemical gradients, which creates electrical currents and voltage changes across the membrane. Ions move along chemical concentration gradients when they diffuse passively from an area of their higher concentration to an area of lower concentration, and along electrical gradients when they move toward an area of opposite electrical charge. It is ion flows along electrochemical gradients that underlie all electrical phenomena in neurons. Changes in the transmembrane potential can cause muscle contraction, gland secretion or transfer of information. Neurons use changes in their membrane potential as communication signals for receiving, integrating and sending information. A change in membrane potential can be produced by anything that changes membrane permeability to any ion or anything that alters ion concentrations on the two sides of the membrane. However, only permeability changes are important for information transfer. There are three types of transmembrane potentials depending on the response of the neuron to a stimulus. A neuron at rest, which means it is not being stimulated, the transmembrane potential is called the resting membrane potential. All neural activities begin with a change from the resting membrane potential. There are two types of signals produced by a change in membrane potential: graded potentials, which are usually incoming signals operating over short distances, and action potentials, which are long-distance signals of axons. Action potentials can be triggered graded potentials, but the action potential does not decrease with distance from the stimulus, and once generated it will propagate along the axon's membrane axolemma toward its termination at the synapse. The potential difference between 2 points can be measured using a voltmeter. The potential across the membrane of a neuron membrane potential is measured by sticking a microelectrode inside the neuron, and the other in the interstitial fluid. On average, a neuron at rest has a potential difference of about -70 mV. The minus sign indicates that the cytoplasmic side of the membrane is more negative compared to the outside for reasons we have discussed previously. This resting membrane potential is designated as VR, and the membrane is said to be polarized. The value of VR varies from -40 mV to -90 mV in different neurons. The resting membrane potential is generated by differences in the ionic makeup of the intracellular and extracellular fluids and the differential permeability of the plasma membrane to those ions. Recall that the cell cytosol contains a lower concentration of sodium and a higher concentration of potassium than the extracellular fluid. The sodium-potassium pumps expends ATP to maintain the concentration gradients of sodium and potassium across the membrane. Of all the many other solutes found in both the cytosol and interstitial fluid, potassium plays the most important role in generating the membrane potential. Potassium is about 75 times more permeable than sodium. The terms depolarization and hyperpolarization are used to describe membrane potential changes relative to the resting membrane potential. Depolarization is a reduction in the membrane potential. The inside of the membrane becomes less negative, meaning that it moves closer to 0 mV. Even minor changes in polarity, such as a change in VR from -70 mV to -65 mV, is a depolarization. Conversely, when a stimulus causes the membrane potential to increase, meaning becoming more negative than VR, this is called hyperpolarization. As mentioned previously, graded potential are short-lived localized changes in membrane potential that can be either depolarizations or hyperpolarizations. These changes cause current flows that decrease in magnitude with distance from the stimulus. Graded potentials are called graded because their magnitude varies directly with the strength of the stimulus. The stronger the stimulus, the more the voltage changes and the farther the current flows. Fluids inside and outside the cells are good conductors, and current, carried by ions, flows through these fluids whenever voltage changes occur. In the picture above, a stimulus was applied to a small patch of the membrane which became depolarized, resulting in a change of polarity at that point. Notice the flip-flop in positive and negative charge at the point of the stimulus shown by the red area. As positive ions flow toward negative areas and negative ions flow toward adjacent more positive areas, local currents are created that depolarize adjacent membrane areas and allow the wave of depolarization to spread. Because the current dissipates quickly and dies out with increasing distance from the site of the initial depolarization, graded potentials can act as signals only over very short distances. Graded potentials are essential in initiating action potentials, which are the long-distance signals. An action potential AP is a dramatic, brief reversal of membrane potential with a total amplitude change in voltage of about 100 mV, seen in the diagram, a change from -70 mV to +30 mV. Notice that there are several phases to an action potential. Following VR, a stimulus was applied that opened sodium channels. As the axon membrane is depolarized by local currents, the sodium channels open and sodium rushes into the cell. Notice the slight decrease in polarity. The inside of the membrane is becoming less negative and is approaching -60 mV. This voltage is called threshold. It is the membrane potential that, if reached, will lead to the generation of an action potential. Step 2 is the depolarization phase, where there is a massive increase in sodium permeability and the transmembrane potential reaches +30 MV. Step 3 represents the repolarization phase. The membrane is returning to its polarized state. The inside of the membrane is becoming more negative. Step 4 shows that the membrane becomes temporarily hyperpolarized, more negative than the resting membrane potential. Notice that there is an absolute refractory period and a relative refractory period occurring during the action potential. During the absolute refractory period, a new stimulus, no matter how strong, cannot trigger another action potential from this neuron. During the relative refractory period, it is possible to trigger another action potential, but it takes a much stronger stimulus than the stimulus that led to the current action potential. The following slide will explain what causes the various steps of the action potential and the causes of the absolute and relative refractory periods. In the resting state 1, all gated sodium and potassium channels are closed. This is the resting membrane potential. The appropriate stimulus causes sodium channels to open, and if enough sodium enters the axon to reach threshold, and action potential is generated. Sodium rushes in, making the membrane potential more and more positive. Notice that the potassium channels are slower to open and close. During repolarization 3, the sodium channels are closing, becoming inactivated, but the potassium channels are open. The result is that no more sodium can enter the axon, but potassium, also a positively charged ion, is leaving the axon. Potassium leaving the axon results in the membrane becoming less positive, it is repolarizing. Step 4, hyperpolarization results because some of the potassium channels are slow to close, and so positively charged potassium is leaving the axon. This temporarily makes the inside of the axon more negative than its normal resting membrane potential. When a patch of axon membrane is generating an AP and its voltage-gated sodium channels are open, the neuron cannot respond to another stimulus, no matter how strong. This period, from the opening of the sodium channels until the sodium channels begin to reset to their original resting state, is called the absolute refractory period. The absolute refractory period ensures that each AP is a separate, all or none event and ensures one-way transmission of the action potential. The relative refractory period is the interval following the absolute refractory period. During the relative refractory period, most sodium channels have returned to their resting state, some potassium channels are still open, and repolarization is occurring. During this time, the axon's threshold for AP generation is elevated so that a stimulus that would normally have generated an AP is no longer sufficient. However, a really strong stimulus can reopen the sodium channels that have already returned to their resting state and allow another AP to be generated. Typically, strong stimuli cause more frequent generation of APs by intruding into the relative refractory period. Recall that the action potential of a single neuron is an all or none phenomenon. It either happens or it doesn't. If a stimulus is not strong enough to cause enough sodium channels to open, then the membrane voltage does not reach threshold, so no action potential is generated. Once threshold is reached, it doesn't matter how much you turn up the stimulus, the action potential will not get any bigger. What does happen as you increase the intensity of the stimulus is that more action potentials are generated in the same period of time. So, stronger stimuli don't result in stronger action potentials, but as stimulus strength increases, the frequency of AP generation increases, more APs per unit time. Let's now talk about action potentials, which are the signals that travel long distances through excitable cells. Only certain cells have membranes capable of generating and propagating action potentials, specifically, neurons and muscle fibers. An action potential is a rapid, brief reversal of membrane potential. It shifts by about 100 mV, moving from -70 mV to +30 mV. Unlike graded potentials, action potentials do not decrease in strength with distance. They are all or none events, and are only generated in axons. This property allows neurons to transmit signals efficiently across long distances without losing intensity. Action potentials follow the all or none principle, they either happen completely, or not at all. When the membrane depolarizes, meaning sodium ions flow into the neuron, if this depolarization reaches the threshold level, an action potential is triggered. Increasing the strength of the stimulus beyond that threshold won't make the action potential any bigger. The size and shape remain the same. However, a stronger stimulus will cause more frequent action potentials, increasing the rate at which impulses are fired, not their individual strength. This frequency coding allows neurons to signal how intense a stimulus is without changing the amplitude of the action potential itself. Once an action potential is initiated, it propagates or travels along the length of the axon. At time 0, the neuron is at its resting potential, around -70 mV. When a region of the membrane depolarizes, shown here at 2 milliseconds, sodium ions rush in, making the inside of the membrane positive. This local depolarization triggers neighboring voltage-gated channels to open. By 4 milliseconds, the depolarization has moved further along the axon while the original section begins to repolarize and return to resting potential. This wave-like process continues in one direction, ensuring that the action potential does not move backward. In other words, once started, an action potential is self-propagating and continues along the axon at a constant velocity, allowing signals to travel efficiently across long distances. Conduction velocity is a measurement of how fast action potentials can travel along an axon. Conduction velocities of neurons are variable, ranging from 100 meters per second to only 3 meters per second. There are two factors that affect conduction velocities of neurons, one the diameter of the axon, and two the degree of myelination. The larger the diameter of an axon, the faster it can conduct action potentials because larger axons have less resistance to the flow of local currents, so adjacent areas of the membrane can be brought to threshold more quickly. There is also less ion leakage because of the surface to volume ratio of the axon. Myelination is a form of insulation. Myelin prevents almost all leakage of charge from the axon, which allows the membrane voltage to change more rapidly. Also, current can pass through the membrane of a myelinated axon only at the nodes of Ranvier where the myelin sheath is absent and the axon is bare. In unmyelinated neurons, propagation of the action potential is continuous, meaning that the voltage-gated channels in the axon's membrane are immediately adjacent to each other. This is analogous to walking heel to toe along a straight line. It is relatively a slow process. Recall that myelination in the CNS is accomplished by oligodendrocytes, and myelination of axons in the PNS is done by Schwann cells aka neurolimocytes. In either case, a sheath of myelin insulates the axon and greatly speeds up conduction velocity. This diagram illustrates AP conduction in a segment of a myelinated axon. Notice that the AP jumps from node to node. Essentially, all the voltage-gated sodium channels are concentrated at the nodes. This type of jumping conduction is called saltatory conduction. Saltatory conduction is about 30 times faster than continuous conduction. In our walking analogy, saltatory conduction is like jumping along a straight line compared to walking heel to toe. A peripheral nerve consists of axons from many neurons. The entire nerve is wrapped in a CT sheath called the epineurium. The epineurium projects inward, dividing the axons into bundles called fascicles. The CT in this location is called the perineurium, and it brings blood supply to the axons within the nerve. Each axon is then covered by the endoneurium. The point is, a nerve consists of myelinated and unmyelinated axons, and axons of different diameters. So when a peripheral nerve is stimulated, the sum of all the AP from all the axons results in a compound action potential. Therefore, although each individual axon of a nerve follows the all or none principle, they may all have different thresholds and conduction velocities. When you run your neurophysiology simulation using PhysioX, you will be stimulating a nerve, not a single axon, so you may see an increase in the AP when you increase the amount of current you are applying to the nerve.